Hi, welcome to our channel Wherever. Today we are going to discuss about the basic structure of immunoglobulin. Just stick to our topic. Okay, first of all, a small introduction about immunoglobulins. At first, immunoglobulins are indicated as Ig, that one is immunoglobulin or antibodies. You see, I is taken from the word immune and G is taken from the word globulins. Okay, immunoglobulins are indicated as Ig or antibodies. Now, what are antibodies? Antibodies are the molecule which are produced by our body in order to kill the microbes. Don't forget what are antibodies. Antibodies are molecules which are able to kill the microorganisms. Then, immunoglobulins are glycoproteins. Chemically, it is made up of glycoproteins. You see, this one is formed in response to an antigen. When the antigen enters in our body automatically produces large amount of antibodies in order to kill the antigen. Next, these immunoglobulins are found in serum, body fluids and tissues. You see, most of us we are confused about whether serum and plasma same or not. You can see this picture. You see, plasma and serum totally differ from one another. You see, the liquid part with the fibrinogen is known as plasma because fibrinogen can able to clot. Okay, so a liquid part, the liquid part with the fibrinogen is known as plasma. The liquid part without fibrinogen is known as serum. Don't forget. Then you see body fluid. Body fluids means using blood, then cerebrospinal fluid, gastric juices, etc. Then you see tissues. Tissues means bone marrow, muscle of various. Then a spleen, lymph nodes, everything coming under tissues. Then these immunoglobulins are synthesized by B lymphocytes. There are two types of lymphocytes, B lymphocytes and T lymphocytes. B lymphocytes are responsible for antibody-mediated immunity. Antibody-mediated immunity. And T lymphocytes are responsible for cell-mediated immunity. Then, these are the small introduction about immunoglobulins. Next, structure of immunoglobulin. You see, the basic structure of immunoglobulin was proposed by Rodney Porter. How to remember this name? Just you see Rod. In the letter immunoglobulin, in the word immunoglobulin, the first letter starts with I. Just it looks like a rod, right? Okay, like that. Rodney Porter proposed the basic structure of immunoglobulin. Next, immunoglobulins are Y-shaped molecules. All immunoglobulins are Y-shaped molecule. You see, it has a stalk, just rod-like structure, and also has two arms. You see, two arms just like our arms. Okay, so immunoglobulins are Y-shaped molecules. The next one is, you can see the picture of the basic picture of immunoglobulin, structure of immunoglobulin. You see, at first antigen binding site. At first the antigen binding site. Here FAB indicates fragment of antigen binding site. Here the short one is light chain, the long one is heavy chain, and the FC region indicates fragment of constant region. Then you see here hypervariable region. The foremost end of the end of the immunoglobulin is hypervariable region that can change according to the molecules of antigen. Then the next one is the variable region. The blue color indicated as constant region. Next you see tip of the limbs are known as antigen binding site. Here when the antigen enter in our body, actually this antigen can react, the antigen can bind only at the antigen binding site. Then tip of the stalk, this one is the stalk region. Here tip of the stalk is known as fragment of constant region. There is no reaction or no structural changes happen in fragment of constant region. The only function is present the whole setup on phagocytes or complements. You see, phagocytes are nothing but cell eating. Complements means these are the complementary system which can able to clear all antigens in our body. Then there are two forms of immunoglobulin. First one is soluble immunoglobulin and another one is bound immunoglobulin. You see, Soluble immunoglobulin always found in dissolved condition, and bound immunoglobulin always bound on the surface of the B cells. You see, the bound immunoglobulins are otherwise known as surface immunoglobulin because it present on the surface of B cells, or membrane bound immunoglobulin. S indicates surface, M indicate membrane bound immunoglobulin. Then you see the surface immunoglobulins which are found on the surface of the B cell is known as B cell receptors, that means BCR. A single B cell can contain nearly 1 lakh BCR on its surface. Just imagine every single B cell contains 1 lakh B cell receptors on its surface. Then you see chemical nature of immunoglobulin. Actually, we already knew that immunoglobulins are made up of glycoproteins. You see, it is made up of 2 pairs of polypeptide chain. That means 4 polypeptide chain. In our body, hemoglobins are made up of 4 polypeptide chain or 2 pairs of polypeptide chain. Then you see, like totally 2 types of chain. One is light chain and another one is heavy chain. In this light chain has 2 chains. You see here one chain and here another. Immunoglobulin has 2 light chains. These light chains are shorter than heavy chain. These light chains are shorter than heavy chain and or identical. Identical means if in this side if it has 214 amino acid, means here also it should have 214 amino acid. This one is identical nature of immunoglobulin light chain. You see, then each light chain is made up of 214 amino acids. How many amino acids? 214 amino acids. This one is very important question. Based on the amino acid sequence, light chains are grouped into K type and L type. K type means kappa type, L type means lambda type. You see, in the serum, 60% of the immunoglobulins are K type and 40% percentage of the immunoglobulins are L type. When you hear K type means 60%, L type means 40%. Then you see heavy chain of immunoglobulin. The heavy chain is made up of 450 to 700 amino acids. This one 214. Here also 400 to 700, 450 to 700 amino acid. There is a flexible region in the heavy chain. That region is known as hinge region. Don't forget, sometimes they ask which one is the flexible region in immunoglobulin. That means the hinge region. Then the main part is disulfide bond which are present in the immunoglobulin. You see, these 4 chains, 2 light chains, 2 heavy chains are interconnected by disulfide bond. Disulfide are also connected in 2 kinds of chain. First one is interchain and also intrachain. What do you mean by interchain and what is intrachain? Interchain means bonding between 2 limbs. Interchain means bonding between 2 limbs. Intra means bonding in the same limb. Bonding in the same limb. You see, 2 heavy chains linked by 1 to 13 interchain disulfide bond. These are the 2 heavy chains. These 2 heavy chains are interbond with 1 to 13 interchain disulfide bond. You see, then each light chain is linked to heavy chain with a single interchain disulfide bond. Here one single disulfide bond, here one single disulfide interchain disulfide bond. Then you see light chain has 2 interchain disulfide bond. You see, wait, light chain has 2 intrachain disulfide bonds. Intra means it is present in the same limbs. 1, 2. So the heavy chains that contains 4 intrachain disulfide bonds. Just you see 1, 2, 3, 4. So first we should know about what is inter and intra, then it is very easy to understand. Then heavy chains are again classified into 5 types. That one is IgG, IgA, IgM, IgD, IgE. IgG means gamma chain, A means alpha chain, M means mu chain, D means delta chain, E means epsilon chain. When you hear the word IgG, that and you should recall this is the only immunoglobulin which crosses the human placenta. That one is from mother to baby. Then IgA alpha. This is the, IgG. This immunoglobulin IgA rich in milk. That one is especially colostrum. Colostrum is nothing but yellow fluid, like yellow fluid. That one we can get this colostrum at the time of delivery or after birth. The mother's milk contains large amount of IgA. That milk is known as colostrum. Then IgM, that one is the mu chain. This one is the first antibody to appear in primary immune response. When the antigen enter into our body, the first immune response indicated immune response is the appearance of IgM. I trust you understood the clear idea about the structure of immunoglobulin. Thank you for watching.